So I'd like to invite our second speaker, Emily Falk from the Department of Psychology, University of Michigan, and she will talk about linking social neuroscience data with health and policy relevant outcomes.
All right, so thanks for the introduction, and thank you for having me here, to the hosts and the staff and the postdocs. I'm so excited to get to be part of this interdisciplinary group. And in terms of what I'm going to present today, I'm going to just give an overview of how our lab has been thinking about addressing these interdisciplinary methods to the question of behavior change. Now, as may be the case for many of us in the room, one of my lab's core motivations is the idea that the leading causes of death are largely preventable. So tobacco use, for example, overweight and obesity for older adults, and then for younger adults, fatal crash. And so as we try and think about how to address these issues, we come up with interventions and programs and policies that are designed to influence individual behaviors, right? Try and get people to quit smoking, try and get them to be more active, try and get teens to do fewer risky things in the car and elsewhere. But in service of a bigger picture goal, right? When we're thinking about a public health approach or how we're going to affect the health of the population, we're thinking about things like, in aggregate, the percentage of smokers who attempt to quit or who start smoking, or the percent of adults who are obese or overweight, or the number of deaths that we're seeing due to motor vehicle accidents. But as social scientists, we know that if we're going to try and move these things, right, either at the individual level or at the population level, it's actually pretty hard to predict the effect of intervention. So in our lab, we think about a couple of different levels of analysis. One is when we think about responses to things like persuasive communications or other forms of social influence that we know impact these outcomes, we can think about individual behavior change. But then we can also think, just like I showed you with those bigger picture maps, about population level outcomes. But predicting these outcomes, as I said, and as the social scientists in the room can tell you, is a pretty tricky task. So research in psychology and other disciplines has given us some reasonably good tools for doing this, right? So we know that constructs like attitudes and intentions predict some variance in behavior change. For example, a recent meta-analysis suggested that on average, constructs from the theory of planned behavior can predict about a quarter of the variance in what people do. But that still leaves a lot of variability left to explain. And so I think one of the exciting things about a conference like this is that there are lots of different ways that we can think about addressing that. And what I'm going to talk about for the rest of my time this morning is one of the ways that our lab does this. And that's to say, can knowing what's going on in people's brains as they're being exposed to different kinds of social stimuli, things like media, things like peer influence, can these neural signals tell us something about individual behavior change and then potentially population level outcomes that's hard to learn from other methods on their own? So in a way that complements the existing measures that we already have. And then third, for the purpose of an audience like this, I thought it was really important to call attention to the fact that when we're dealing in these basic science mechanisms, my hope is that the knowledge that we get from the kind of work that I'm going to talk about can be used to facilitate understanding not just the brain and psychology, but also designing better interventions, programs, and policies. So as just a brief roadmap for where we're headed today, since this is an interdisciplinary meeting and I wasn't completely sure about the prior expertise of the audience with respect to neuroimaging, I'm going to spend just a little bit of time talking about functional MRI, which is the primary method that my lab uses to extract these neural signals. And then what I'm going to do is talk a little bit about some of the earlier work that my colleagues and I did to try and figure out whether the brain could get some traction on predicting individual and population level behavior change. And then given that we're thinking about lifespan issues and particularly focusing on adolescence and then older adulthood, I'm going to talk about some recent data from my lab at Michigan where we're looking at recently licensed teen drivers and social influence in a context where we're not looking at neural responses to mediated communications, to health communications, but instead to more basic social, cognitive, and emotional processes and how that complements the rest of the work that we're doing. So as I said, the primary method that my lab uses is functional MRI. And in a typical MRI study, we had a question about this earlier, kind of what does this look like? And so in a typical MRI study, we have participants lie in a machine that looks like that for about an hour, as Kevin said. And while they're in the scanner, they can do pretty much anything you could have them do on a regular laptop, right? So they can see things, they can see pictures or text or videos, they can hear things through earbuds, and they can make responses using different kinds of response collection devices. And as they're doing these things, just like you might have them do in a behavioral lab, we send out these safe radio frequency pulses that excite protons and water molecules, right, in our blood. And as the protons relax, that tells us something about what's going on in different parts of the brain. The relaxation rate in different parts of the brain tells us something actually about the changing concentrations of oxygen. And this gives us what we call the blood oxygen level dependent signal, the BOLD signal. So, one take-home point from this is we're looking at this proxy for neural activity over the course of a time where people are doing things that we manipulate psychologically. And then what comes out is essentially a big 3D movie of brain activity over time. And so in order to do the analyses that we want to do, we divide the brain up into these little 3D pixels that we call voxels. And when we follow a voxel over time, we can construct a time series, just like you'd be familiar with in other kinds of behavioral research. We can apply similar statistical models where we look at the variation of the BOLD signal according to some model that we've specified with respect to what we think we're manipulating psychologically. We can then engage in the same kinds of parameter estimation and hypothesis testing that you might with other kinds of work. And I say this just to say that even if you're not an imaging person, right, the same kinds of statistical questions apply, and then we deal with the special things with algorithms. And one of the special things about imaging data is since we have so many different voxels that we're looking at, sometimes we display, as you saw in Kevin's talk, these visual images of the outcomes of those statistical tests, right? And we call these statistical parametric maps, where often the outcome that we're interested in is the brain activation. So we regress, say, some kind of individual difference or neural activity onto the brain activity, and the brain is treated as the outcome. But one of the keys to understanding the rest of what I'm going to tell you today with respect to how I've been thinking about these processes and the way that our lab treats the brain data is that there's now a really nice mass of information about where different things tend to take place in the brain. So the kind of decade of social cognitive neuroscience plus cognitive neuroscience data gives us a really good idea of where we might start now to think about the brain not just as an outcome, but also potentially as a predictor variable that we can include in models. And so we can take the information that we've gotten from brain mapping studies, and we can also take what we know from social psychology, cultural psychology, health psychology, about how different psychological processes relate to real-world outcomes that we're interested in. And then we can directly hypothesize relationships between those brain mechanisms and outcomes that we care about. So, as I noted, when we think about the brain as a potential predictor of outcomes, we can think about it predicting individual behavior change, which I'm going to talk about, and then also potentially these more aggregate or population level outcomes. So the first study I'm going to tell you about, I'm going to start out going a little bit chronologically and how we were thinking about this work when I was in grad school. And the first example of this brain as predictor approach I'm going to offer is using sunscreen, changes in sunscreen use as an outcome variable. And we started out with a really basic question, which is, if we have some hypothesized regions of interest within the brain, can we predict behavior change outside of the scanner at all? Right? Is this something where we can construct models where there's a significant relationship? And then if we can do that, are we explaining the same things that other measures are explaining, or are we explaining different things, right? Can we explain above and beyond what we're getting from other sources? And so in order to attack this question, we developed a paradigm in a domain that has been successfully used in lots of health psychology research, namely sunscreen use. And in this study, we had participants look at messages like these about the need to wear sunscreen while we monitored their neural activity. And in addition to monitoring neural activity while they were being exposed to these different kinds of messages, we measured their reports of attitudes towards sunscreen use, their intentions to wear it in the week following the scan. We had some information about their behavior in the week preceding the scan. And then after they were exposed to the messages, we collected these same kinds of measures. And then there was a surprise follow-up 1 week later, where we looked at how much they had changed their behavior then from the week prior to the scan to the week after the scan. So this is our measure of behavior change in this kind of low-stakes domain where we thought we could get some traction.
Can we see the message again?
Sure. So there were a number of different kinds of messages. This one reads, "Baby soft skin, yours again. Would you like your skin to be baby smooth and clear into your old age? According to dermatologists, the most reliable way to promote healthy, soft, fit skin is to protect your skin from the sun." And it goes on like this. So these are very standard messages. These we took from the American Dermatological Association, I think. And what we're looking at is basically aggregate responses to these. So as opposed to work that I can talk about in the discussion period where we're trying to tease apart sort of differences between messages, here we're just looking at sort of overall response to an intervention. Okay, and so to set up where we looked, in this framework where we're using the brain as a predictor, we have to say in advance, where do we want to look? And so the starting point here was the medial prefrontal cortex, which if you divide the brain in half is sort of here in the middle towards the front. And this has been implicated in a number of different things that we thought were relevant, for example, self-related processing. The very specific studies we focused on were the ones that we thought were closest to what we were going for, so studies of kind of persuasion and intention. Brian's also done a lot of really important work looking at the role of ventromedial prefrontal cortex that Kevin alluded to, and this is actually something that I think is really interesting and exciting, and we finally have a little bit of data for me to speculate about what's going on that we can also talk about in the discussion. But this is where we started as a guess for where might we look in the brain to see if we can predict something afterwards. And in particular, we constructed these little cubes, these regions of interest, based on the coordinates from relevant studies. And then we measured neural activity in the regions of interest during exposure to this intervention. So what I'm showing you on the x-axis here is our predictor variable is neural responses in this region of interest. And then the outcome that I'm going to show you here is behavior change after we've partialed out variance that's accounted for by the self-report measure. So controlling for attitudes and intentions. And so if there's something that the brain is explaining above and beyond what we get from other sources, we should see a positive relationship here. And indeed, that's what we see. And we explain about an extra quarter of the variance above and beyond the self-report measures that we collected. So I don't know how excited you guys are about sunscreen, but I was super excited when we got these initial results because from a method standpoint, right, it really suggested that we could do things with neural activity that weren't just looking during the time that the scan was taking place, but also we could project out into meaningful behavior change in the real world. And so in order to kind of build on this work, what we next did was run a whole brain search. So looking throughout the brain, beyond these regions that we had started out with, and we looked at what regions were then most highly associated with behavior change. So it's basically just going the other way with the regression. And what we found was within medial prefrontal cortex, you can see now we're no longer dealing with a cube, but instead a more refined region that was the part of medial prefrontal cortex that was most highly associated with behavior change in the context of this sunscreen study. So we have one group of participants where we've now defined a new region of interest, which we can carry forward into other studies that I'll tell you about now. So the next thing that we did, right, this conference is about leveraging interdisciplinary methods to understand behavior change. And as I have kind of alluded to, sunscreen is important, but it's not the most motivationally relevant behavior that you can think of, although that sunscreen work was done in LA, and so it's a little different than Michigan, for example, or Berkeley. We were really interested in whether the results that we found in the sunscreen study would then also extend or generalize to a context where we have a more motivationally relevant health behavior, and then whether we could push the time horizon out a little bit. So in order to look at that, we recruited a community sample of smokers who were motivated to quit. So again, since this was part of the early program of research, we didn't want to have too many moving pieces. So we held constant their stated intentions to quit and their stage of change. They had to be taking action to quit, as evidenced by having signed up for a quit program. So we partnered with Freedom from Smoking in LA to recruit these folks before they started to try to quit. And what we recruited were smokers then who were relatively heavy smokers. They were active smokers at the time that they were recruited, but as I said, were held constant in terms of their stated intention to quit. And then the methods I'm going to show you here are in form very similar to what I showed you for the sunscreen study. So in particular, we collected a number of baseline measures, but we expanded the set of measures now to not just include attitudes and intentions, but also self-efficacy and ability to relate to the messages. We got a slightly more robust set of behavioral indicators, so not just self-report, but also with smoking you can get expired carbon monoxide, which is a proxy for recent smoking. Participants then did the ADS task, which I'll tell you about in a minute, while we monitored their neural activity, and then they started trying to quit. And we collected a lot of really cool data that I'm not going to have time to go into right now, where we did experience sampling for 3 weeks. But what I'm going to focus on is the change from their baseline smoking to their endpoint. So again, we have this measure of endpoint smoking compared to baseline smoking. And in order to develop this ADS task, we partnered with a number of different organizations. So another thing that I think folks in this room are probably more comfortable with than a general audience is the idea that by working with community organizations and other kinds of folks who have different expertise, we can get really cool stimuli that then ultimately have real-world data tied to them. So here we partnered with the Louisiana, California, and Massachusetts Departments of Public Health, as well as the American Legacy Foundation. And they gave us ads that they had developed for our target audience, which is smokers who are trying to quit smoking. And all of the ads across the different campaigns had basically the same sort of general theme, which is we know it's hard to quit, but there are resources that can help you quit, and here are some resources. So to give you just a little bit of a sense of what these ads look like, I wanted to show you basically two ends of the spectrum for conveying this message. So this first one is basically a cutesy version of we know it's hard, but here's some resources. So that's one end of the spectrum in terms of an approach. Here's a more serious version of that same message. Right, so as you can see, there were a wide range of approaches in terms of ad content, but the basic message was we know it's hard and here are some resources, namely the National Cancer Institute's Quit Line, 1-800-QUIT-NOW. And so our hypotheses for this study basically involved replicating the effect that we had seen in the sunscreen study. And in particular, we looked at neural activity in this functionally defined region of interest, right, which was defined based on the sunscreen participants. And we looked within that region as participants were then exposed to these smoking ads. And an open question was then, will the relationship that we see hold after we control for these additional self-report measures, right? So for things like self-efficacy and ability to relate to the ads. And what we found was that neural activity in this region of interest was significantly associated with behavior change. So the change in smoking from baseline to a month later, both on its own and controlling for each and all of our self-report measures. And to quantify basically this variance in terms of how much we're explaining in the outcome, if we start out with just intention and self-efficacy, we explain about 9% of the variance in behavior change. If we add in this self-relevance component, basically how much could you relate to each of the different ads, we explain about 15% of the variance in behavior change. And that self-relevance measure was inspired by the idea that maybe it was some kind of explicit self-related process that was going on in MPFC. And then if we look at the MPFC activity on its own, what we see is that we get about 17 or 18% of the variance. And when we put them together, they're actually independent of each other. And so we approximately double the variance that we can explain in behavior change. So this particular outcome that I'm focusing on is the expired carbon monoxide levels. We also had various self-report measures like 7-day point prevalence. So here we find that activity in this a priori region of interest within medial prefrontal cortex doubles the variance that is explained in that outcome that we were interested in. So that speaks a little bit to the robustness of this finding across individual behavior change. But one of the things, one of the reasons that I started doing this work in the first place was I was doing some work with organizations trying to make health information and services more accessible to young adults. And I was really interested in whether we could construct effective health communications that would work for the greatest number of people. So one other way that we could measure success here is, can we predict something at the population level? Now, that would involve linking the neural activity in a relatively small group of people, say our group of smokers, to a larger group of people. And so a way of asking this question is, does, for example, neural activity in the same region of interest that predicted individual behavior change also index the behavioral responses of larger groups of people? And so by partnering with these different public health agencies, I'm going to show you data from the same sample where I showed you their individual behavior change. We focused on 3 of the campaigns, which are each made up of multiple ad exemplars, that ended with NCI's quit line, 1-800-QUIT-NOW. And I was saying by partnering with these organizations, we were able to get measures of quit line call volume in the month prior to the launch of the campaigns and then in the month following the launch of the campaign. So in some ways, we can look at changes in quit line call volume that are attributable to the launch of these campaigns. And it was convenient for us that in the places where the media markets where these were tested, there weren't other tobacco-related media on the air. They had relatively comparable, for example, levels of taxation and so on. So for simplicity, when I describe these, I'm going to refer to them as campaigns A, B, and C. And in setting up the ads task, I showed you exemplars from 2 of them, from campaigns B and C. And when I showed you those ad exemplars, probably if you're like most people, you kind of mentally compared them, thought about which one you thought might be effective or less effective. And that's something that we actually also had our participants do after they got out of the scanner, but for each of the ads within each of the campaigns. And so you can kind of think about 2 different types of, say, focus group measures. One are the typical kinds of things that we might get around a table, right? So things like, how effective do you think these ads are? And we had our participants rank order the ads in terms of most effective to least effective. We also had them tell us their most favorite to their least favorite. For each ad, we also had them complete a 10-item scale, which we developed based on items from the Legacy Media Tracking Survey and other measures that are typically used to evaluate the success of these campaigns. And these all give you the same answer, essentially, in terms of how effective the different ads are. And then the other way we can think about potentially evaluating the different campaigns is by looking at, just for the simplicity of this study, the average activity within medial prefrontal cortex, within this region of interest that we were focusing on. So as I said, across the various self-report measures, we got the same answer. So here I'm showing you the data from the self-report rankings of effectiveness, but the picture looks the same for the other self-report measures as well. And in particular, what this picture tells you is that participants essentially liked campaign B the best and campaign C the least. Then the second way that I was saying we can think about ordering these different campaigns is in terms of the activity that they elicit within that region of interest that predicted individuals' behavior change. And here we see a different pattern of results. So I'm showing you means here. And in particular, right, the placement of campaign C is opposite. So we get a pretty different prediction about the ordering of these campaigns depending on whether we look at average activity in this region of interest or whether we look at participant self-reports. And
can you just remind us which one is C, which one is B of the ones?
Sure. So C is the finger puppet ads, which people didn't like at all. And the one that they liked the most was that the campaign where I showed you the guy smashing the copy machine. It's designed to be rather empathic, and a lot of the participants could relate to that campaign, and that's what they told us afterwards, at least. Mm-hmm.
So the implicit contrast in the imaging, is it just rest?
Yeah, it's compared to rest.
Sorry, it's just the absolute magnitude of activity compared to rest?
So yeah, basically what we did here was we looked at just modeling each of the campaigns on its own compared to rest. So it's not, say, percent signal change or something like that. So when you're thinking about, the unstandardized coefficients, it's a little bit tricky to interpret. And here, given there are a lot of limitations to this study that I'm happy to talk about in the discussion if you guys want. But another thing to think about is that here we're just focusing on also essentially ordering, right? So I'm showing you means, but we did a lot of other statistics, non-parametric tests, and other ways of assessing basically, if you look at all different possible orders that people could have come up with, either within subjects or across these different things, is this something that you would expect to see by chance or not? This is actually a nice example of the peer review process, because initially we just had these means and we were like, isn't this interesting? And then the reviewers sort of pushed us to dig deeper, and I think it strengthened things a lot.
I just clarify one more thing. So was the scan really short, or did they see the ads multiple times? Were there just the 3 ads interspersed with red?
No, so what I was saying is I showed you an example from each of these campaigns, but each of the campaigns actually consisted of 3 or 4 ads, and then they made ratings after each one. So they saw each ad one time, and so the task takes about 11 or 12 minutes.
So when you say C, that was multiple ads that were...
Exactly. They all have these finger puppets who are in different kind of social situations. And the copy machine guy is one exemplar from a campaign where they have kind of, another one involves a woman who's on the phone and it's raining out and she jumps out of a window to pick up a butt on the street. And they are these kind of serious ads that are designed to acknowledge what smokers are going through as they're trying to quit. So what we found when we looked at this picture is that there's something potentially different about the self-reports and the neural activity. And as just one more sidebar, in some work that we're doing right now, I think it's not necessarily a question of either or, but how do we optimally combine? But in this situation, given the small number of ad exemplars we had and so on, what we did was we looked at these 2 different kinds of rank orderings. And then we looked at what happened at the population level. So changes in quitline call volume from the month before the campaigns were aired to the month after they were aired. And here's what we see. They all increased quitline call volume, but some did so more than others. And in particular, the pattern that we see is parallel between the neural activity in this region of interest and the population level outcome. Or just visually, here's what they look like. So to take a step back and just consider the overall picture of these first couple studies that I showed you, the data suggest that neural activity in a subregion of medial prefrontal cortex can predict not just simple health behaviors, but also motivationally relevant health behaviors like smoking. and that this also seems to map onto important population level outcomes that we care about. For example, people calling a quit line. And there are a number of different approaches that my lab is taking right now, that, as I mentioned, that are designed to do a couple of different things. One is kind of elucidate what the psychological mechanisms are that are producing this effect, right, so that we can translate them back to measures that we can disseminate more widely, and also because we care about the basic science of it. And then also, you know, Emily had asked me a little bit earlier about some of the cultural moderators of these effects, so we're also looking at boundary conditions on the effects that I'm showing you here. And I'd love to kind of get a chance to talk about this, but given the theme of the conference in terms of emotion regulation and lifespan transitions, and especially adolescence, I wanted to just take the remaining formal talk time to tell you a little bit about some data that we've been collecting for the past year and a half or so that focuses on, back to the individual level, still using the brain as predictor approach, in ways to understand how we can understand social influence in a way that may not be apparent with just self-report measures alone. So the next study, instead of focusing on neural activity in response to mediated communications, in response to health communications, looks at what's going on in people's brains as they're being exposed to more basic social, cognitive, and emotional tasks. And I'm going to just focus on one kind of social emotional task now. And this is work that's in collaboration with some folks at NICHD and the University of Michigan Transportation Research Institute. And we're focusing on recently licensed teen drivers. So we're particularly interested in changes in risk-taking. I think Kevin did a really nice job setting up some of the reasons why we might think about adolescence, and I think that's probably near and dear to many of you in this room. And in particular, there are lots of different ways that risk-taking can manifest itself in adolescence. We focus on driving decisions because fatal crash is the leading cause of death in teens between 16 and 19 years old. And peer influence can be especially dangerous for teen drivers. So teen drivers are at elevated risk for fatal crash when they have passengers in the car. So when we think about the major risk factors for fatal crash as cited by the CDC, we have being recently licensed, we have being male, and then, as I said, having passengers. And in particular, adolescents driving in the presence of peers are about 5 times more likely to be involved in fatal crash. So there's something really interesting that we may want to consider with respect to this idea of social influence, right? I was saying the lab is really interested in understanding neurocognitive and emotional predictors of influence and understanding how those work. So basically, this is just a way of saying that this important health and policy issue also speaks to what I think are some important theoretical questions in social psychology. And in particular, what are the interactions between contextual and individual difference factors that lead to increased versus not increased susceptibility to social influence in this period? And within the brain as predictor framework that I've been talking about today, can the brain tell us something about the potential mechanisms that lead some teens to be particularly susceptible to social influence? And of course, this builds on lots of work by folks in this room and others who have examined the neural bases of risk-taking and social processes in this group. I was saying to Ron earlier, it's been really fun for me to start to get into this area of understanding adolescence because it's not something that was part of my formal PhD training, but that the lab is doing more and more of, both with respect to these kinds of questions that are health relevant, but also because we're really interested in things like message propagation in the new media environment. And so that's another thing we can talk about in the Choose Your Own Adventure. So, as Kevin also mentioned, right, there's this idea in terms of when we're thinking about why adolescents take more risks, that it may be explained partly, at least, by asymmetric development in the brain. So we see heightened emotional responses and slower development of cognitive control systems that regulate these. And this kind of effects of the imbalance are especially exaggerated in social situations, right? So this is a kind of perfect segue here. And then also, as Kevin alluded to a bit towards the end of the talk, right, it's probably actually not the whole story to just say emotional versus cognitive, right? We really need to consider social context as well, right? So social context can mean a lot of different things. In the data I'm going to talk to you about today, I'm going to just look at being alone versus being in the presence of a peer. And then there are individual difference factors in terms of how people respond to equivalent social inputs. So in terms of hypotheses, when we were thinking about understanding susceptibility to social influence in recently licensed teen drivers, we thought about what might predispose people to be more susceptible to risky social influence, to risky peer influence. And we hypothesized that increases in neural activity within brain regions that in other contexts have been characterized in terms of social pain or the distress of exclusion, that this might be associated with increased susceptibility to this risky influence because it might be more costly for people to be excluded. And so if it's more costly for you to be excluded, then you might preemptively take steps to fit in. And so this behavior that looks kind of maladaptive, right, doing things that potentially are going to kill you, taking risks in a car, may be adaptive to the extent that it's more costly for you to be left out. Now, in particular, to set up the kind of specific brain as predictor approach we used, we looked at neural activity in an imaging lab in some specific regions I'll tell you about in 1 second. And then a week later, we looked at susceptibility to risk-taking in a driving simulator lab. And I'll also give a little bit more detail about that in a second. And then we collected a number of different self-report measures, like things like susceptibility to peer pressure, resistance to peer influence, impulsivity, and a number of other things. And in the brain, what we focused on were regions that in other studies of teens in particular have been found to be associated with this experience of distress during exclusion. So the subgenual cingulate and the anterior insula. And for folks who are familiar with, say, Naomi Eisenberger's work in this regard, the results I'm going to show you are substantively the same whether we include the dorsal cingulate or not, but it doesn't seem to drive these effects. And Carrie Maston actually doesn't get dorsal cingulate when she does this exclusion task I'll set up in a second in kids. So the participants for our study were recruited by the University of Michigan Transportation Research Institute, and they got us people who are in this highest risk demographic, namely males who are 16 to 17 years old and who had gotten their type 2 driver's license in Michigan within the past 4 to 9 months. So this is the license that allows them to drive on their own, and that's when we see that spike in risk for fatal crash. Then in the scanner session, what they did was they came in and they were introduced to 2 gender-matched Confederates, so 2 young men, and they were told that they were going to be playing some computer games together and then doing some other tasks on their own. How many people are familiar with cyberball? Okay, great, so a lot of you, right? So you know there's a phase where the participant is included, so they see the 2 other Confederates' names on the screen and they throw the ball to each other and everybody gets the ball approximately equally. And then in a second phase of the game, our participant is excluded. So the other people stop throwing the ball to them. And the design here, just to give you a bigger picture view from the participant's standpoint, is first they do this imaging session where they do cyberball and some other tasks, for example, go, no-go, things that are trying to get at this tension between emotional responses and regulation. And then 1 week later, they go to UMTRI, to the Transportation Research Institute, and they drive in the simulator alone and with a passenger. The order of these effects is counterbalanced, and I'm going to control for it in all the analyses I show you. And in particular, we used a Confederate as our passenger to hold constant who's the person in the car with them. Then within the driving simulator session, they drive on a predetermined course, and we focused on their behavior at yellow lights. So one of the advantages to working these kinds of interdisciplinary teams is the driving experts tell me, you know, what's a really good proxy for risk-taking in the real world when we're dealing with these complicated data from the driving simulator, and we settled on behavior at intersections that have yellow lights. And in particular, driving through a yellow light, being in the intersection when the light is red, is the risky behavior, and stopping is the safe behavior. So just a quick recap here. The predictors that I'm going to focus on are some self-report measures like susceptibility to peer pressure, and then neural activity, the average neural activity in the social pain network during the exclusion phase of cyberball compared to the inclusion phase. In all of the analyses I'm going to show you, I'm going to control for drive order. There's also another way that we manipulated the confederate behavior that doesn't interact with the effects I'm going to show you, so I'm just going to control for it here, but we can also talk about other ways of conceptualizing context effects in this situation. And then the outcome that I'm focusing on, as I said, is this risk-taking at intersections. So to remind you of the hypotheses, right, here we thought that increases in activity in this social pain network during exclusion compared to inclusion should predict increased risk-taking in the presence of a peer compared to driving alone, and that this should be selective for the social situation, right? We think that the social context matters here. So we shouldn't see this exclusion compared to inclusion activity predicting solo risk-taking behavior when there isn't a peer around. And the idea here, again, is this costly exclusion idea. And then, again, we have the open question of whether we'll explain similar variants or different variants according to things like susceptibility to peer pressure. So do participants know how likely they are to change their behavior when they're alone versus when they're driving with a peer? And so are we getting the same or different variants? So what I'm going to show you results-wise, then, is the effect of just social context on its own, being with a peer versus being alone in the simulator, how their reports of susceptibility to peer pressure predict their behavior when they're driving with a passenger. I'll then review kind of the fMRI analysis, going to Bob's point about trying to keep everybody on the same page, and then show you the results for how the neural activity predicts the outcome that we're interested in before and after controlling for our self-report measures, and then also show you this kind of selective effect. So consistent with lots of research on teens, we found that teens behaved in a riskier way on average when they were driving with a peer compared to when they were driving alone. So they blaze through more yellow lights, or they're in the intersection during the red more when they're not supposed to be with the peer compared to when they're alone. And interestingly, we found that their self-reports of susceptibility of peer pressure don't actually predict this effect when we control for the various experimental setup.
I'm sorry.
Yeah.
What was the Confederate doing at the time?
So the Confederate manipulation is pretty minimal when they're in the car. So the Confederate pretty much just sits in the car. Before they get in, I was saying we manipulated some characteristics of what the Confederate is like, and there was a norms manipulation beforehand where they watch some videos together, and the Confederate makes different ratings depending on the between subjects condition, and then they actually get to see the Confederate drive before they drive in the spirit of fairness, right? So everybody gets a turn in the sim. And so for the fMRI analysis, we look at neural activity during exclusion compared to inclusion in the subgenual cingulate and anterior insula, and then we use that as a predictor of their risk-taking behavior during the passenger drive. And what we find here is that when we examined the neural activity, increased activity in the social pain network, as we hypothesized, was associated with increased risk-taking in the presence of a passenger, controlling for their solo risk-taking behavior and the various other covariates that I described. And this also holds when we control for susceptibility to peer pressure. So we're getting at different constructs here. And then finally, as we suspected, this is not the case when we look at solo drive behavior. So it seems to be selective for the social situation. So just to summarize what I've showed you so far with these teen passenger data, we found that increased activity in our a priori hypothesized social pain network during exclusion compared to inclusion predicted driving risk in the presence of a peer, which was selective for that social situation. And this is above and beyond some standard measures we have. So I showed you the susceptibility to peer pressure measures, but this also holds when we control for lots of different individual difference factors that we might be interested in, and is consistent with this costly exclusion hypothesis that I suggested, which tells us something about how we can think about using this brain as predictor framework to also address hypotheses about social psychological processes, potentially.
Clarifying question.
Sure.
You have scanner data both for the driver and for the passenger, right? Because they...
No, we just have scanner data for the driver, and the passenger is a confederate. So we hold the passenger behavior and actually the passenger person constant across all of our drives, right? So the scanner session happens at one time point, and then the driving happens a week later. So taking a step back in terms of the various data that I've showed you so far today, across these different kinds of health behaviors and situations, we find from a method standpoint that we can combine neuroimaging with other kinds of measures to predict additional variance in outcomes that we care about. And more and more what the lab is interested in then is trying to pin down what the psychological meaning of those activations should be. So what does this tell us about psychology at the individual, group, and population levels? And then finally, I wanted to basically point out what all of you know, which is that to conduct these really large-scale studies, it's a big team effort. So for the teen passenger study, there have been a number of graduate students and staff involved, in addition to collaborators at UMTRI and NICHD. For the smoking data that I showed you so far today, a number of folks at UCLA, Matt Lieberman and Elliot Berkman in particular, are really instrumental in thinking about this. There's a lot of money involved, so I wanted to acknowledge people who are funding work we're doing now, and then thank you for having me here and being part of this discussion.
First of all, thanks for the introduction to the neuro... Oh, we have lollipops. Thanks for the introduction to the neuroimaging. I thought that was really helpful and lovely and kept us all involved. So I was wondering if you could go back to the basic finding in the first half of the talk, where you have this part of the brain, it's sort of sitting along this little piece of brain that's telling us something about subsequent behavior, health-promoting behavior, above and beyond all the psychological things, efficacy and whatnot. And could you talk about that part of the brain in the kind of an intimate, descriptive way as to what it usually does, what you think it's doing, why that activity might have this sort of powerful prediction of subsequent behavior? And I think you talked about your excitement and that kind of finding, where you're going from a little sliver of tissue to a complex behavior at a different point in time is really very unusual. So I thought maybe you could tell us what that little sliver was all about.
Yeah. So Bob's question was about sort of what is the function of the medial prefrontal cortex in general, and then what do I think it's doing here? And the tricky thing, right, is that what it does in general isn't something that I can answer in a super concise way, because just like all of the rest of the brain, there's a one-to-many mapping between brain activity and then different kinds of psychological processes. And so from a method standpoint, we're moving more and more towards considering networks and patterns within regions, but I will answer your question. So. Yep. So we started with this region because it's the region that's most frequently implicated in studies of self-related processing. So doing things like answering whether an adjective is me or not me, compared to whether it's printed in uppercase font or lowercase font, right? So a self-related process or not a self-related process. And that's why we started there. And I think that was actually a lucky guess, but for the wrong reason, right? Because that's an explicit kind of self-related process. And then we found we could predict the variance. But then when I go and explicitly ask, can you relate to this ad, it doesn't mediate the relationship. It's also, as I said, in Brian's work and other people's work, implicated in this process of evaluation. So for example, Antonio Rangel's lab has done work looking at when you're evaluating, say, a food stimulus, and you have different dimensions like healthiness and tastiness, and you're trying to integrate those. You see activity in medial prefrontal cortex doing some of that balancing act of that valuation. So I think that's something that may be involved. But the thing that we're pursuing most deeply right now is the idea that you can have self-related processes that aren't explicit of the type, do you relate to this? Is this me or not me? And so in a study right now where we have some funding from NCI, we're looking at physical activity behavior. And I wanted to do a couple of different things. There are a couple of pieces that are missing from what we've done so far. One is we don't have in these prior studies any kind of implicit or indirect measures. So in this next study, we're building in a couple of different reaction time implicit measures that look at connection between the self and behaviors that we're interested in. Another thing we're doing is, can you manipulate? So far, I haven't manipulated anything, really. Can you manipulate self-related processing? And one way that you can do that with respect to self-related processes is something called self-affirmation. Is everybody familiar with self-affirmation, right? So the idea is that if you give people an opportunity to affirm themselves in an unrelated domain, that it allows them to take other information that in a non-affirmed context would be self-relevant and threatening, and to process it as more self-relevant. So in this physical activity study we're running right now, we have participants who are sedentary, and I'll just give you a little schematic for what we're doing here. So we have stimuli that were designed in conjunction with the Michigan Center for Health Communication Research. And we have people come in and we train them to use an accelerometer. We get a baseline week of accelerometer data. They then do this intervention, and we get a month of accelerometer data afterwards. And so we can calculate the same kind of behavior change measure as before, but during that scan session, we have a between subjects manipulation of being affirmed versus not being affirmed. And what we see is, in terms of preliminary results, that over time, activity in this same medial prefrontal region that I've been focusing on seems to be associated with decreased sedentary behavior. And here's the cool part that starts to get at the psychological mechanisms. We've developed a couple of what we call localizer tasks, where we identify regions of the brain and independent tasks that are associated with processes that we think should matter. So we've developed what we call a counter-arguing localizer, where participants are given statements of the form people should, and then we pre-tested the heck out of a whole bunch of different things to have things that are easy to argue in favor of and easy to argue against, and then hard to argue in favor of and hard to argue against, so we can balance the difficulty of this process. And they argue in favor and against different things, so we can identify neural regions that are active when people are counter-arguing statements that aren't about health. We can also look at neural activity during self-related processing in this kind of simple self task. And then we also have the neural activity during the affirmation manipulation. People still with me? Okay, awesome. So what we find is that activity during the affirmation, this affirmation effect, is associated with increased activity during the messaging task, during exposure to health relevant messages about physical activity compared to control messages about balancing your checkbook or other kinds of things. And that activity is associated, for now, we just have proximal outcomes like changes in self-efficacy, and that it's positively associated with that. And that the affirmation effect is negatively associated with lateral prefrontal regions that show up in our counter-arguing localizer. And these things are all kind of interrelated. And so what we're working on right now is basically putting the pieces together to say, we have some preliminary evidence that when we manipulate this self-related processing, we see increased activity in medial prefrontal cortex and actually also in the kind of precuneus posterior cingulate region that we've focused on is also part of this self network. that it's affected depending on what happens during the affirmation manipulation, and that the way that people are using the part of the brain that's showing up in our counter-arguing localizer is also affected. So that's where we are right now with respect to, does that get a little bit of what it might be doing? So some possibilities are evaluation, some kind of implicit self-related process, something that's tied to the regulation processes that Kevin touched on, right? So the kind of tension between counter-arguing versus absorbing the information. And we're in the process of trying to unpack some of those things a little bit more.
Do you think that, I mean, it seems like there's individual differences. You're showing that there's individual differences in how people respond to the intervention that then in medial prefrontal cortex that then predict later behavior. Do you think these individual differences are specific to the stimuli, the smoking stimuli, or the, whatever you're presenting? Or do you think you could find some unrelated task, even during resting state scan, does medial prefrontal cortex activity relate to how they respond to an intervention and how much they're likely to engage in it? So could you predict the same things using medial prefrontal brain activity outside the intervention to how they'll respond to the intervention?
Yeah, so that's something that we've been thinking about a lot in terms of, are we getting at something that's specific to influence, to social influence? Or is it something about people's, say, receptivity to social stimuli or something else like you're alluding to? And the short answer is I don't know yet. We have been collecting some resting state scans and considering that possibility. The other thing we've been trying to do then is manipulate, with small sample sizes, the only thing that's really feasible to do are these within subject manipulations. But if you start to tease things apart with between subjects manipulations, this is a different route than you're suggesting, but I think yes is the first part of your question. It's important to know. And then with respect to the between subjects things, we can look at, with a control task or something else, what do we see? And so, I think it's a really exciting and important thing to know, and we have some pieces in place to try and get at that, but I don't know yet.
One interesting comment is that one thing that argues against this from your data is that you used rest as the baseline, and you're getting the effects in contrast to rest. So that would kind of argue against my hypothesis here, that
Maybe.
If it's people
so I agree with respect to the kind of default activation of medial prefrontal cortex and using rest as a contrast there. In the physical activity study, we have rest and a resting baseline, and then also these other kinds of messages that we compare to. But I think in the early studies, this comes up sometimes about is it something related to the messages per se, where I think the population data are the strongest data about it being maybe something about the messages, right? But I think there's also an individual difference component where we're maybe reading out, even though people we hold constant people's intention to change or where they think they are because they're going and signing up for a quit program. But there may be something else that speaks to how successful they're going to be at doing it.
Okay, so let me stop for a second. So the long-term goal that we have in the Behavioral Change Research Network is to integrate different perspectives from different fields and to try to develop a unique approach to the study of behavior change. But I think that first we need to understand the other perspectives. And for me, I'm guessing that for you too, this is very far from what we are economists are doing. The method, the neuroimaging is very far from what we're doing, even though there is a new field, neuroeconomics, but it's not widely accepted in economics, and most of us don't know a lot about it. And the other thing is emotion regulation is a term that I first heard a year ago. We don't know anything about it. And I think this all are things that we can learn about, but we first need to understand how it's related to how we think about stuff. And so I would like to start with a question to you, Kevin, about what you're doing in the lab with emotion regulation. So we see people that you instruct them to do this exercise, regulate their emotion, and I'm wondering, you see some region that is activated. Is it related? Do we know how much it's related to what happens in real life when it's event that actually relates to them, something that happens to them? So first, you will not instruct them to regulate their emotions. So what do they do then? And the other thing is that it's actually concern them. So for example, the distraction strategy that you talked about, is it actually relevant in real life? We can distract for a moment, but if it's related to me, I will come back to that event and will think about it again. So what is the relationship between what's going on in the lab and real life?
I'm glad we started with a small question. Such an easy one to answer. Boy, gosh, I suppose there's a lot of different things to say. So I can start off by saying that we're always interested in understanding the extent to which what we ask people to do in the lab reflects what they do in everyday life. And we wouldn't be doing what we're doing if we didn't think it did meaningfully reflect that in some way. I think one major difference, just conceptually, is that while people engage in reappraisal, like the example I gave from the aftermath of Superstorm Sandy, people won't explicitly represent it for themselves as I'm reappraising. People commonly find or search for alternative ways of framing things, or we have these aphorisms like look on the bright side, or every dark cloud is a silver lining, and we're advised to do these things by our friends. And we may attempt to do it, but we don't necessarily think that we're regulating our emotions. And we don't represent it to ourselves in the same way we would as scientists thinking about someone from the outside. That said, people like Oliver John, he was here a moment ago. Is he still here? Maybe he left. And James Gross, they developed a questionnaire to measure the frequency with which people say that they use reappraisal in everyday life. And they've related that to various types of longer-term sort of mood or physical health outcomes. So the frequency with which people report reappraising in everyday life does predict things in a beneficial way. It doesn't necessarily relate to what we ask people to do in the lab for an interesting reason, however. So at least it typically doesn't. So if you look at the frequency with which people say they reappraise in daily life, it doesn't predict how well they do on our tasks typically, because we train people up and we give them a very explicit context when you should do it now, don't do it then, do it now, don't do it then. Rather than having people in their daily life be free to choose when they're going to do it and seeing how effective they are in that context. We actually there's a student in the lab interested in when people choose to reappraise and how effective they are if they've been given the choice to do it versus being told to do it. And he's sending me data, I think, even as we speak right now. Also, we have a lot of work in clinical populations that I didn't talk about at all. We have collaborations with various groups at Columbia, at UCLA, at Mount Sinai, looking at reappraisal abilities or other abilities in clinical populations, where the question is, if there's something real about the regulatory abilities we're testing in the lab, it should predict dysfunction or be related to response to cognitive behavioral therapies, where we think reappraisal is sort of an in-the-moment thin slice of what you're doing in a cognitive behavioral therapy in a longer term. And it looks like we've got very encouraging results from those contexts, that it looks like it does tap into those things.
It sounds like if you knew more about how emotion regulation affects decision-making, then you could measure better what's going on in real life. You could actually observe the results. So what do we know about how emotion regulation affects the decision-making?
So that's an interesting question. And I think there's a little bit of work on that coming out from Antonio Rangel's lab, but I've long thought that a lot of decision-making tasks are de facto emotion regulation tasks, because in the act of making a choice to delay gratification, you're regulating your emotion in some way. Like, I'm going to go ahead and wait and not respond to the initial impulse to consume something right now. That reflects sort of a down weighting on the value of something in the present moment in favor of some more abstract representation of its value for me in the future. And so, and some of the early studies of temporal discounting, it looked kind of like some of the same brain regions you see for other sort of cognitive control tasks in general, when people would sort of delay, or reappraisal even. So it's something where you're re-representing the meaning, which normally you just respond to in a sort of bottom-up hedonic kind of way. You re-represent it in a more abstract format and use that representation to guide your behavior. That's kind of the essence of top-down control. And people do that in the process of making decisions all the time, although again, they don't represent it to themselves as I'm regulating my emotion as I'm doing this. It doesn't mean it's not emotion regulatory, however.
you think that the food example is the most, it's decision making and emotion regulation, it's both.
Yeah, I think depending on what hat you wear, you might call it different things.
Yeah, we have some, with the teen passenger data that I presented, the cyberball part of it, we also collected go, no-go data, which measure response inhibition, which isn't emotion regulation, but the brain systems that are involved in emotion regulation overlap with motor response inhibition. And so the rationale there was if we know something about the teens, what's going on in their brains when they're doing this basic response inhibition task, that might tell us something about their risk-taking decisions in the driving context. And we find support for the hypothesis that sort of the interaction between the neural activity and the social situation according to the Confederate behavior predicts risk-taking.
Yeah, I feel obligated saying something given that I'm sitting here. And then I think we'll give it back to the audience. So let me first, it's going to sound that I'm very grumpy. So let me first criticize economics before I'm...
Before you criticize us.
So look, I think that, economist view, the traditional economics view about behavior change is that let's give people the right incentives. If I'll give you enough money, or I'll change your incentives by taxing you enough, then you will change behavior. If people are eating non-healthy food, we need to give them the right incentive to eat healthy food. And my view about it is that we definitely failed. So this is for economics. The problem of behavior change is much more complicated and multidimensional than just giving people the right incentives and they will take it from there. This is a very Chicago economics approach. But now for us, I think, for the problem for an economist like me to take what you guys are doing and actually using it. So I think that the first problem is that we talk about behavior change. And for me, even self-reporting behavior is not behavior. And even going further, calling 1-800 to the helpline is not changing behavior. I just called the helpline. It doesn't mean that I changed my behavior. So I think we, at least I'm as an economist, I want some assurance that when we are saying changing behavior, we really mean that we change behavior. I'm always giving the example that if you ask me if I like dolphins, I will say sure. If you talk with me about flipper, I will tell you later on, this will be the manipulation, that I like dolphins even more. But if you track how much money I gave to dolphins, this will be zero throughout. So in some sense, you didn't change my behavior towards dolphins in any way. So this is the first point. The second point, I think that there is something that makes economists, it's difficult for economics to come on board. I think it's the differences in statistical methods. So I can tell nothing about neurometrics, if even this field exists. I look at this picture and I say to myself, I don't understand any of this, it must be wonderful. So I'm not questioning in any way your statistical methods. But I think in actually taking the brain data into the field and looking at the correlations, I think here actually economists can help. Many people are, I see in these talks, they are putting a regression which is linear, and then they say, it's not in this talk, and then they say, look how linear it is. Yes, of course it's linear. You are running a linear regression. And the relationship is highly non-linear, actually. So I think that, economists, for better or worse, we became the statisticians of the social scientists. We tend to focus maybe on completely uninteresting questions and really study them very precisely. But here I think that there is a little bit of, I don't want to say that we have different standards, but we definitely use different tools. Now, finally, for me, probably, I'm asking for you to do something that I cannot. So, in an ideal world, I would like, suppose Ayala and I, we both smoke. I want you to put us in a scanner and to say, Ayala is a good decision maker, so we can give her information about smoking, and this will be enough to change her behavior. Shachar is a bad decision maker. Tell me from the scanner. So even if I give him a lot of information about smoking, he will not be able to basically digest this information. We need another intervention with this guy. So basically the question is, can scanner data help us identifying whether it's preferences, information, beliefs, or constraints? I'm sure it's a combination of all of them. But I think solving the identification problem is extremely important if we talk about policy. And just to give a concrete example on this, for example, teens driving. You know, my daughter is 10, and she's a teenager. I just spoke with a co-author, and I told him that my daughter is 10 and she's already a teenager, and he said my daughter is 30 and she's still a teenager. So we are talking about a very long here. And you know, it's hard driving with her. She talks constantly, she distracts me. So in some sense, I can stop and say, you know, enough, I just can't handle it anymore. Probably her peer will not say it. So in some sense, I think about teens driving. It's not that there is another teen here, he makes me take more risks. He's actually changing my preferences. I really think about my preferences as something very constant. I think it's a constraint. It's a constraint having a teen in the car. They just drive you nuts. And finally...
And the epi data say that if you look at sort of who's being risky in the car, like if you follow people out of high school parking lots, so male drivers with male teen passengers are doing risky things, but so are adults with teenage passengers. It's just that the adults aren't getting in as many fatal crashes.
So, and finally, just to wrap it up, let me even say something about, you know, terminology. You know, we keep, you know, even in the conversation that we have here in Berkeley, we say emotion, emotions, emotions. You know, for me, after so many conversations, for me, emotions are preferences. So not information, it's not beliefs, it's not constraints. Maybe I'm wrong, but I think it's preferences. And I view preferences as something very stable. And I think that you guys, exactly, I know,
He's
okay.
wrong, that's okay.
That's fine, that's fine. I feel that changing someone's preferences, especially, you know, adults, this is very hard. Preferences are very strongly held. And whenever I see that you are changing emotions, you know, it brings me back to my first point. Yeah, self-reporting preferences you can change. But true preferences that are governing behavior, I feel that it's very hard to change. So I don't know where to put the question mark, so I'm continuing. So maybe my question is emotion preferences? How, translate it to me, please, because as you can see, I'm confused and a bit lost.
So that was a lot. Do you want to start there, or do you want to cycle back to the beginning? And
Yeah, I mean, I think there are a lot of interesting questions in there. One is, I completely agree with you that we want to integrate brain data with a number of different kinds of measures, including what people perceive, because that's important, and then also what they do. So right, I was saying, different slide. With our physical activity data, we have accelerometers on people. We're measuring what they actually do. With the smoking, we had expired carbon monoxide. So that tells us at least some proxy for how much recent smoking they've engaged in. And so I agree with you that in some situations, self-report is not a good predictor of behavior, and so we can measure behavior, and we do. In terms of the methods question, part of the reason that I wanted to give that quick overview of imaging methods is to say, you know, even though you may think of imaging as being really different from what you do, we're using in the sort of vast majority of imaging studies you'll see, it's based on the general linear model. It's the same kinds of basic statistics that you're familiar with a couple of tweaks to account for, you know, the lots of voxels that we're looking at. That said, there are some economists or neuroeconomists, people who span multiple disciplines. Greg Burns and Brian's lab. You know, basically, there are ways that you can take these kinds of models and apply them to brain data plus other kinds of data that we care about. And so I think the issue of methodological sophistication or the kinds of models that we choose, you're completely on the same page, I think, with where our field is moving. Yeah, in terms of the question about whether you pay people and they change their behavior, there are other kinds of incentives that are at our disposal too, right? So things like social norms. And so I know various folks here, right, you were saying you've been looking at some of the ways that we can think about this, and A lot of our labs have been interested in social reward and social context as something that's an incentive to behave in a certain way. In our lab right now, we're doing some work besides the individual level and the population level. If you think about what connects those, there may be some element of message propagation or, you know, passing information from one person to the next. And so we've been looking at that partly because it's interesting scientifically, but then also partly because if you can shift culture, if you can shift norms, I think you're wrong that these preferences remain very stable. And there are lots and lots of social psychological data that tell us that the context matters a lot.
And about the mechanism, if you can say something about the mechanism, when you see an active area in the brain, do you learn whether it's the information that was persuasive or what's going on there?
The short answer is no. The longer answer is you can start to try and get at more information by doing things like I was describing, where you localize activity in regions that are associated with a process that you think is important, or you look at the literature and make hypotheses, right? Just like with any other psychological research where we operationalize a construct in some way, right? We ask certain questions, or we observe behavior in some way, or we do something which is a proxy for an underlying thing that we care about, right? We can think about the brain in this way also. And so we make hypotheses and we test those hypotheses and we try and rule out as many other explanations as we can. And then, you know, if the brain data from, say, a brain mapping study generate a new hypothesis, we can try and then come up with clever ways to test that in, say, a behavioral lab context. And then the last part of your question about emotion and whether emotion equals preferences, I'm going to mostly defer to Kevin on this since he's the emotion regulation expert. But you know, one of the things that that we've been thinking about more and more in the lab is, I showed you the costly exclusion data. A lot of the data looking at teen decision-making or risk-taking focuses on the reward system, and there have been a handful of social influence studies now that have focused on the ventral striatum and the brain's reward system as predictors of social influence. And so I was saying to Kevin right before I gave this talk, I had a version of the talk where the overarching framework was basically social influence conceptualized as emotion regulation, right? Basically these individual difference and contextual factors that push around that dimension. And so, so there's part of that I agree with, and the stability part I would take issue with. And I think you are probably the best person to speak to the rest of it.
Yeah, unconfuse me.
I guess I would just first ask you a question. What do you mean when you say preference?
Okay, that's a very good question. When I say preferences, I mean the ability to solve trade-offs. And I'm not saying in which way you solve trade-offs, but for me, someone that has preferences, it's someone that is able to solve trade-offs in a coherent way, in the sense that you're going to give him a set of options, and he's going to be able to order this option from the very best to the very worst. And what I believe that if you give someone a set of options, and regardless of the domain, and I don't want to say whether it's my belief or my hope, maybe it's kind of wishful thinking. My hope is that if you give me the same set of options, I will be able to order them, in the sense that I will never sit with two options and I will say, I don't know how to rank it, or I will have cycles in this order. And hopefully, I call preferences if the way that I order it on Sunday is exactly the same as the way that I order it on Monday, and the noise in my order is not coming from really my underlying preferences, is coming from a constraint. For example, my preferences about taking an umbrella. If you see me, and you see me one day taking an umbrella, and another day don't take an umbrella, it doesn't mean that I like the umbrella less. I like it exactly the same. But there is just, I'm not facing the constraint of the weather, so I'm not taking the umbrella.
What about framing effects, though?
Yeah, so for example, before framing effects, whatever you call norms or social influence, I will put it all in constraints. So, my preferences is to have green hair. That's just not the social norm in the economics department. So the fact that I don't have I don't paint my hair in green says nothing about my preferences. It just says that I have a constraint. It's not a monetary constraint. It's a social constraint. My colleagues are going to look at me in a weird way, and I'm going to be penalized for it. So I think that when you are saying emotions, you, in my language, you mean that this preference ordering that I have is always changing. And it will even change if I'm holding everything else in the environment, the information, the beliefs, the same. I will always have it different.
I don't think I'm saying that. I think we mean different things when we talk about preference and emotion, if I'm understanding you correctly. If I take your meaning of the word preference to be that there's some, as revealed in my behavior, some stable way in which I'm going to make choices to engage in this action, but not that action. Or when given a choice between two actions, I'll tend to engage in A before B reliably and consistently. I'd say you're describing a particular pattern of choice that may be stable over time. And that's what you mean when you say preference. When somebody who's an affect scientist uses the term preference, they mean a sort of a valenced evaluation. Something is good or bad that may or may not influence the kinds of preferences that you talk about. It could be that I really like A and I don't like B, and that's part of why I reliably choose to do A and not B. But it could be that I really don't like A and I really do like B, but your situational constraints dictate that I always have to engage in A. Like, I like to speed and drive really fast, but I have to drive the speed limit. Crap, okay. I don't like doing that. It's irritating to me, but I'm going to do it because I have to. So your emotional responses, or your affective preferences, if you will, from an affective science perspective, aren't the same as the kinds of stable patterns of behavior, what you're talking about. They may be related, they may be unrelated, it may depend on the context, may depend on the constraints that are of interest. And I think one salient point for people who study emotion is that, like the old adage, a rose by any other name would smell as sweet, is just not right. Because your emotional response to things is a function of a number of different factors. And so it's like the old Pepsi challenge experiment, right? If you know that you're drinking Pepsi versus Coke, you prefer Coke because it has greater cultural cachet and coolness. But if you don't know what you're drinking, you prefer Pepsi. So that drink by some other name did not taste as sweet to you. And in that sense, there's all kinds of variables that are influencing what our emotional response is to something at any given moment. And in that sense, emotions are not stable properties necessarily of an interaction between an individual and an object, because the nature of the interaction can change as a function of context, as a function of time, as a function of my internal context, my mood, my motivational state, and so on.
And you said something about training, right? That I can train myself to...
Well, if you're interested in emotion regulation, then you can train yourself in a variety of ways to probably have an enduring response, affective response to a stimulus that's different than it was initially. So that's partly what's of interest to us, and some of the smoking research that we've done is if you ask people to effortfully attempt to change the value that they place on smoking a cigarette, to change the craving that a cue will elicit for them, you can measure simply behaviorally in the lab their ability to do a reappraisal-like task that uses cigarette cues that elicit cravings, where they think about negative long-term consequences versus short-term immediate hedonic consequences. By getting them to focus on the negative long-term consequences, you can just measure their self-reported change in craving on day one, and their ability to show decreases in craving in that task will predict 2 months down the road how much they actually decrease smoking. So there's something about these tasks, you asked about the real world, that predicts their ability to do these things.
This is like a magic. I don't understand how it predicts the real world outcomes after a long time.
Well, we can certainly talk about more. I don't think it's magic at all. I think the thing that, the variable that's often left out of the kind of work that we're doing in my lab, but we're trying to build in more and more, is a critical one, which is, once you bring people into the lab, you can tell them, here's what you would do to regulate your craving in the moment. They can show that they can do it effectively in the lab. They know what the strategy is, but when they go out in their daily life, it either doesn't occur to them in the moments where they would need to regulate, that they should be regulating now, or if it does occur to them, they're distracted by a bunch of other things. And so the really critical question is kind of, how do you harness their motivation to actually implement a strategy they know will be helpful? And how do you make sure it's delivered to them at the moments where they most need it? And so we're collaborating with a couple of people at NYU, social psychologists named Peter Gollwitzer and Gabriel Oettingen, who study kind of self-regulatory techniques that more or less provide an automated delivery system of self-regulatory strategies in the context in which you need them. And they have a technique for sort of identifying what goals people want to accomplish, making sure you're motivationally committed to pursuing those goals, and then formulating simple if-then statements where there's an if context, like if I'm in the following context, which is the trigger for the need to regulate your behavior in some way, then here's the thing I'm going to do. And simply formulating things in an if-then way is kind of magic in a way that we don't understand yet, but we're trying to. Doing some brain imaging studies that then allows you to sort of automatically engage the regulatory strategy you want in that context. It sort of primes you to have that context elicit the use of the regulatory strategy. And it seems like it's relatively automatic and doesn't require effort to bring it to mind. And so we think that's kind of the and then a miracle occurs step.
There are also individual differences in this. So we have some data. There's a paper that Elliot Berkman is the lead author on from these smokers that I told you about, where we had them do a go-no-go response inhibition task. And then what we find is that neural activity in those experience sampling data that I mentioned, if you look at the slope of the relationship between craving and smoking, in general, that's a positive relationship. But if you look at neural activity during the go-no-go task, what we find is that for people who are a standard deviation above the mean in the kind of stop network when they're doing that response inhibition task, that the relationship flattens out, right? So the relationship between craving at one time point during the day and smoking a little bit later, so craving at noon and smoking at 2 p.m., that goes away if you have this sort of, higher level of activity in these regions during response inhibition task. On the other side of it, if you're a standard deviation below the mean, then the slope is much steeper. So if you crave, you smoke.
So, I mean, one thing I can add to that is that there's something of an idea emerging in the literature that the sort of suite of prefrontally based brain systems involved in self-control can be flexibly deployed across a number of different contexts. So if you're measuring one kind of self-regulatory ability as expressed in a go, no-go task or simple inhibit your response task, to the extent that the same suite of processes is engaged there as is engaged in a reappraisal task, you can predict your ability to perform one from your ability to perform the other. And there may be some tonic way in which engaging in self-control of various kinds kind of tones up these kind of self-regulatory muscles in a way that makes you kind of more of a chronic self-regulator. And that's kind of I think, some of the assumption underlying the kinds of data that she just described.
I think we have to stop because we must see it.
We are losing our credibility. We promised to give it back to the audience. We are already losing our credibility. But it's not the economist's fault. I gave very long answers, these guys. Thank you.